Diet and lifestyle have many impacts on cognitive function and mental health. This video is going to focus on how different nutrients such as lipids and carbohydrates can impact your brain, how your diet can influence your genetics, and how your gut and brain are interconnected. Omega-3 fatty acids are essential polyunsaturated fatty acids. Two types of omega-3 fatty acids are DHA and EPA. DHA and EPA are found in fish or taken in supplement form. DHA and EPA have many important functions in the brain, including building and maintaining brain structures, modulating brain perfusion or blood flow, and reducing inflammation. The omega-3 index is a measure of EPA and DHA in red blood cell membranes. During pregnancy, DHA is transported across the placenta to the fetus to help build brain structures. A lower omega-3 index in the mother is associated with perinatal and postpartum depression, as omega-3s are important in reducing inflammation in the brain, which is linked to depression. Moderate levels of DHA in formula during infancy led to improved problem-solving, attention span, IQ test scores, and language skills in childhood. In children and adolescents, higher levels of DHA and EPA are associated with improvements in cognitive function. A higher omega-3 index is also associated with slowed cognitive decline, improved aspects of memory, reduced, age-dependent, loss of gray matter and white matter, a later onset of dementia, and improvement in Alzheimer's disease in the early stages. The levels of omega-3s in the blood are also related to brain injury, psychiatric disorders, and mental health issues. A low omega-3 index may be a predisposition to psychiatric disorders, as many are a result of improper brain development, such as major depression, bipolar depression, schizophrenia, psychosis, BPD. Lastly, regarding mental health, anxiety is negatively correlated with the omega-3 index, while mental toughness is positively correlated with the omega-3 index. Carbohydrates can be split into two types, simple and complex. Simple carbs combine in varying ways to form complex carbs. Because complex carbs must be broken down before use, they remain in the bloodstream longer and contribute to long-term blood glucose stability. Carbs are extensively involved in cognitive function, as the brain uses glucose as its primary fuel source. Carbs play an important part in regulating brain metabolism, balancing neurotransmitter levels, mood and stress responses, and helping to develop long-term cognitive resilience. Additionally, stable glucose levels are associated with emotional stability. For the brain to receive glucose, it must permeate through the blood-brain barrier. This is because the brain has high and rapidly changing energy demands, but minimal internal energy reserves. Astrocytes, glial cells involved in neuron support, coordinate energy delivery and modulate metabolic balance by regulating glucose distribution. They are also able to store glycogen and mobilize it when brain tissue is under high demands. Low-carb intake is acutely associated with impairments in working memory, attention, executive function, and emotional regulation. Chronically, low-carb intake can increase anxiety, irritability, and mental fatigue. Carbs modulate neurotransmission by influencing the production of neurotransmitters, including serotonin, dopamine, and GABA. Additionally, they modulate the availability of tryptophan, a molecule involved in serotonin synthesis. High sugar intake can disrupt dopamine pathways and induce addictive eating patterns. Carb consumption also influences mental health. Diets high in simple carbs are associated with increased anxiety, depression, irritability, and cognitive fatigue. Alternatively, complex carb diets are linked with improved stress resilience, psychological well-being, mood, and sleep quality, while decreasing neuroinflammation, neurodegenerative disease risk, and cognitive decline. Nutrigenomics is the study of how diet affects gene behavior. Diet and lifestyle influence proteins and markers surrounding DNA, and has been found to influence gene expression in every human being. It does this through metabolism, nuclear receptors, and signal transduction. Diet impacts you from the moment you are conceived, with prenatal dietary effects impacting the development of cognitive functions. Key bioactive compounds impact your gene expression. Positive effects have been found from compounds like omega-3, vitamin D, antioxidants, and B vitamins. Negative effects have been found from compounds like omega-6, simple carbs, and protein energy malnutrition. Epigenetic changes through nutrigenomics can lead to changes in cognitive function. From the moment you are born, postnatal effects can be seen with animals, showing that if they are raised in a relaxed environment with food present, they will likely become a relaxed adult. Along with this, dietary changes like proper omega-3 consumption has been found to assist with frontal lobe and hippocampus function. However, on the negative side, DNA methylation, which is influenced by diet, can result in the development of cancer and non-genetic diseases. Along with this, deficiency in methyl donor micronutrients such as folate, B12, choline, methionine, and B6 during early life can result in altered behaviors and cognitive functions. Nutrigenomic effects can also be found within mental health. An increased risk of having anxiety and depression can be found due to both prenatal and postnatal experiences with stress and vulnerability in food, leading to further likelihood of mental illness presentation. Oftentimes, this stress can be transgenerational, resulting in impaired mental health functioning that can last for generations. And even more specific examples of nutrigenomic impact on mental health have also been found, such as vitamin D deficiency leading to increased risk of autism and schizophrenia, and vitamin B12 deficiency during fetal neurodevelopment leading to oxidative stress, neuropathy, and depression. Gut microbiota are a vast collection of independent bacteria within our gut. They help with digestion, immune system function, integrity of the mucosal epithelial layer, and have antimicrobial properties. It also communicates to the brain via the gut-brain axis. They are integral to our overall health. Normally, these bacteria are stable, but certain aspects of lifestyle, like consuming excess simple sugars, fats, or antibiotics, can throw this balance off. When conditions like this occur and the balance is broken, it is called dysbiosis. Dysbiosis can occur when there are too few bacteria, they are too harmful, or there is little diversity among them. This is able to affect cognition through the gut-brain axis. The gut-brain axis is the name given to the system between the two, connected through the vagus nerve. It is called bidirectional because both sides send and receive signals. The enteric nervous system is a system of neurons that exist in the lining of our guts. It contains neurons that can act independently from the brain and the central nervous system, and communicate with the brain through the vagus nerve, using neurotransmitters. One of the most prevalent results of a faulty gut-brain connection is irritable bowel syndrome. It is caused by improper connections between the gut and brain, whether that is dysbiosis of the gut or improper nerve sensitivity by the brain. Dysbiosis can lead to leaky gut syndrome, which causes widespread inflammation and weakening of the blood-brain barrier. This allows excess neurotransmitters into the brain. An example is choline turning to acetylcholine and others turning to glutamate. Higher levels of glutamate can lead to excitotoxicity, neuron death, and cognitive decline. It can lead to diseases like major depressive disorder and Alzheimer's disease.